feel free to ask me to annotate. All right, guys, so here we are in chapter 13. This Sunday, you just have the Labster Lab do the action potential. And a couple of you were asking me why Canvas doesn't update your score right away. And I'm not sure why that is, but sometimes it can take several hours, maybe even 24 hours for Canvas to update your Labster score. So if you want, you can take a screenshot of your final score, but Labster has always updated Canvas. It's just sometimes there's a lag time with that. And then, so that's the only thing you guys have due on Sunday, I believe, is just a lab. We'll start chapter 13, which goes through the peripheral nervous system and your reflexes. And we include your special sense organs in this. So tonight we're going to focus on the eye. And I've finally done this, and I'm sorry I didn't do it for the previous PowerPoints, but I did chop off a lot of this PowerPoint. I think it originally was 150 slides. So I chopped it down to 68. And even as I'm going through this, I'll try to point out what I want you guys to focus on. So we'll kind of jump right in here and we'll try to get through part 1 tonight, part 2 on Tuesday, and we should be on track. And then eventually we'll be done with the nervous system, because I know you guys probably want to start learning about other body systems too. So we're looking at the peripheral nervous system. We've kind of focused on the brain and spinal cord in chapter 12. In the peripheral nervous system, remember, this is how we break it down into the sensory and motor division. So the sensory is called the afferent division. Motor is efferent or effector division. In the motor division, we have the somatic and autonomic. Your somatic nervous system supplies motor activity to all skeletal muscles, glands. And the autonomic nervous system is under subconscious control. So these things happen automatically. And this can be your sympathetic or parasympathetic division. Remember, your sympathetic is the fight or flight response. So it helps to get your body ready for emergencies. And the parasympathetic is the rest and digest division. And tonight we'll go through the peripheral nervous system, and we'll talk a little bit about sensory receptors, sensation, and then we'll focus kind of the second half, just looking at the eye and how we see. So survival of any organism depends on sensation, which is the awareness of stimulus, which is a change in an internal or an external environment, and then perception of that stimulus, which is the interpretation. And this makes sense that survival depends on this, because your body makes adjustments based on changes in pH, based on changes in temperature, in glucose, and the ability to interpret the meaning of that stimulus and then make a change is how we survive. The somatosensory system is part of our sensory system that serves our body walls and limbs, and it receives inputs from all these different types of receptors: exteroreceptors, proprioceptors, and interoreceptors. The input will be relayed toward the brain, but kind of processed along the way. And we'll go through those different types of receptors, which is what I want you guys to focus on. Adaptation is a change in sensitivity to a stimulus in the presence of that constant stimulus. So let's say you're constantly being bombarded with a stimulus, the receptor membranes will eventually become less responsive. The receptor potentials decline in frequency or stop. We have phasic receptors or tonic receptors, which describes how fast they adapt to this stimulus. And all I want you to know is that adaptation, is when your receptors kind of stop being sensitive to a stimulus. And you guys know that if you're staring at a wall or a dot on a wall, eventually that dot, or in the periphery, starts to kind of disappear because that sensory stimulus that is going into your eyes eventually just stops. So that's what adaptation is kind of the ability to change in sensitivity in the presence of a constant stimulus. Perception of pain we'll go over a little bit. So pain is a warning of actual or impending tissue damage. So protective action can be taken. Some pain impulses are blocked by inhibitory opioids like endorphins. Pain tolerance, all perceive pain at the same stimulus intensity, but pain tolerance varies. So if you are sensitive to pain, it means you have a low pain tolerance, not a low pain threshold. Genes can actually help determine your pain tolerance as well as response to pain medications. So if someone is really sensitive to pain, don't come down on them too hard because it's something that they can't control. And if someone isn't that sensitive to pain, I think of women who give birth naturally over and over again. They might have a high pain tolerance, or they're just tougher than I am. I don't know. So, but that is sometimes related to genetics. Visceral and referred pain. Visceral pain results from the stimulation of a visceral organ receptor. So it's felt as a vague, aching, gnawing, or burning. And this is activated by the tissue stretching, ischemia, chemicals, and muscle spasms. And referred pain is when pain from one body region is perceived as coming from a different region. And a good example of this is when you have left arm pain during a heart attack. How that is occurring is that the visceral and somatic pain fibers, so the fibers coming from the heart, as well as sensory fibers coming from the left arm skin area, they both travel along the same nerves and they enter into the same spinal root. So the brain will assume that stimulus is coming from a common somatic or skin region. And we have areas and maps of referred pain, as this is important to keep in where I don't know if I might ask you one question about what referred pain is. But other than that, you don't need to know, exactly where everything is. It's important to know that if someone comes to your ER and is complaining with left arm pain, that's a telltale sign of a heart attack. But same thing, if someone has a right shoulder pain, there could be something going on with the liver. Lungs and diaphragm is in the upper left region. So anyway, referred pain, the somatic and visceral fibers follow the same nerve. So the brain perceives the visceral pain coming from a different skin or part of the body. So that's referred pain. We have some homeostatic imbalances with referred pain, hyperalegia, which is pain amplification, chronic pain, and phantom limb pain. So due to limb amputation, we can have kind of receptors that are activated by long-lasting or intense pain. Specifically, phantom limb pain can be felt in a limb that has been amputated. So now they use some epidural anesthesia during surgery to reduce that phantom pain. Okay, so sensory receptors are activated by changes in the internal and external environment. The awareness of a stimulus is sensation, and then the interpretation of the stimulus is perception. And there's 3 ways we classify receptors: the type of stimulus, the body location, and the structural complexity. And these are the types of receptors I want you to know. Maybe there'll be a matching question. Maybe you'll have to pick one out of a multiple choice. So by stimulus type, mechanoreceptors respond to touch, pressure, vibration, and stretch. So you'll find a lot of those in your skin. Thermoreceptors are sensitive to changes in temperature. Photoreceptors respond to light energy. So we'll find photoreceptors in the eye, which I'm hoping we get to tonight. And then chemoreceptors respond to chemicals like smell, taste, and changes in blood chemistry. And then nociceptors are sensitive to pain-causing stimuli like extreme heat or cold, excessive pressure, inflammatory chemicals. Nociceptors, which are responsive to pain, can then go on and stimulate other types of receptors, thermo, mechano, or chemo. So those are nociceptors with a star. Then exteroreceptors respond to stimuli arising outside the body. So we'll find these in your skin and also in most of your sense organs. And then internal receptors are also known as visceroreceptors, because viscero stands for organs. So these respond to stimuli in internal viscera and blood vessels. So they'll be sensitive to chemical changes, tissue stretch, and temperature changes. Proprioceptors respond to the stretch in your skeletal muscles, tendons, joints, and ligaments. And they inform the brain of your movements so that your brain knows where your body parts are in relation to each other. So you're not randomly hitting your arms or slapping your face, or your legs aren't kind of hitting each other when you're running. So that's proprioceptors. It's the ability of proprioception for your brain to tell where your body parts are in relation to the other. So then we have simple receptors of general senses like touch, just complete general senses. And then the special senses are usually housed in complex sense organs. And we're going to go over vision, or the eye, today. So then we'll kind of jump into the eye, and the rest of the PowerPoint kind of focuses on the eye. There will be some anatomy review of the eye, and you will have I think maybe one or two matching questions labeling parts of the eye. So you'll want to just spend some time reviewing just the anatomy and the structures of the eye. So some 70% of your body's sensory receptors are actually all located in the eye. The eye itself will convert light energy into electrical signals, which is kind of cool to think about. Light comes into the eye, and then your brain can perceive the images. It consists of accessory structures and the eyeball, and we'll go over some of those anatomy now. So here is the eye and accessory structures, everything that surrounds the eye. Not going to ask you too much about the accessory structures, but you have your eyebrows, your eyelashes, basically things that are trying to keep outside sweat or debris from entering into the eye. So these are some of the accessory structures of the eye. And they're all labeled here. The conjunctiva is a thin membrane that lines the inside of your eyelid. And then we'll go over the lacrimal apparatus and a little bit of the 6 extrinsic eye muscles that move the eye. So again, here's just a look at the eye and accessory structures, including the eye muscles that attach to it, and so forth. Conjunctivitis, again, we have an itis word, which means inflammation. This is an inflammation of the conjunctiva, which is, again, the inner kind of pinkish membrane on the inside of the eyelid. And this can result in a reddened or irritated eye. And pink eye specifically is an infection of the conjunctivis, but it's caused by bacteria or viruses. And if you have kids who've ever gotten pink eye, it's highly contagious. So the lacrimal apparatus consists of the gland and ducts that drain into the nasal cavity. It's what will create tears, and tears contain kind of some mucus, antibodies, and lysozyme, which help to keep the eye very clean. Blinking spreads the tears towards the medial side or towards the nose, and then tears eventually empty out into the nasal cavity. So here's your lacrimal gland secreting tears that kind of wash from lateral to medial across the eye, and then drain down the duct into the nasolacrimal duct into the nasal cavity. So your nasal cavity is constantly getting rid of these tears, but if you're crying a lot, you have excess tears, and that's why you usually hand someone a tissue if they're bawling their eyes out. Here are the 6 extrinsic eye muscles. They're all named for how they attach themselves to the eye, and they all move the eye in different locations. So you can kind of see them here. You have 2 oblique eyes, which are kind of off at an angle from the eyelid itself. And this shows you the movement that they make with the eye. So whenever you're thinking of what muscle and what the action of a muscle is, just think of the muscle shortening and then pulling on that body part. So for example, if your medial rectus muscle pulls on the eyeball and it's located on the medial side, so by your nose, it'll turn the eyeballs inward as if you're kind of going cross-eyed. If the lateral rectus muscle constricts, it'll turn your eyeballs outward to look out to the side. And then I think I have a chart that describes the action of each with the controlling cranial nerve. I don't think I have any questions about specific actions of each muscle. There might be some cranial nerve questions, but no actions about those. Then we get into the structures of the eyeball, and this is where hopefully you remember this from anatomy, and if not, just review this because I do have, I think, 1 or 2 complete labeling questions of the eye. There's 3 layers: the fibrous, vascular, and inner layer. The internal cavity is filled with different fluids called humors, and we separate, the lens separates the internal cavity into the anterior and the posterior segment. So let's take a look kind of at the layers first. We have the sclera, which is the white part of the eye. The choroid is the next layer. It's kind of this pink layer that contains blood vessels. And then the 3rd layer that's yellow is the retina. That's the nervous layer. If we work our way over, the cornea is a part of that fibrous first outer layer. It covers the pupil and the iris. We have the ciliary body, which is the type of smooth muscle that controls the shape of the lens. And then you can see here kind of where we have an anterior segment, which contains aqueous humor, which is more like watery fluid. And then we have the posterior segment behind the lens that contains vitreous humor, which is more jello-like to kind of help give the shape to the eyeball. We have the optic nerve attaching to the back of each eyeball. So that brings in all our nervous stimulation, our nerves, our axons. A central artery and vein of the retina enter at that optic nerve. So right here where the optic nerve attaches and all the axons enter and the blood vessels enter, we call that this is your blind spot, because in this area you have no photoreceptors. And we'll talk about what photoreceptors are, but this is your eye's blind spot. And your eyes are constantly adjusting images so that they don't fall on that blind spot because your brain wants to see an image as clear as possible. So that's why you have 6 eye muscles that helps your eyes constantly adjust images so that the images are not reflected back on this blind spot, because you wouldn't be able to see them if they are. Also on the retina, we have an area called the macula lutea. And within that, there's a little tiny pinpoint called the fovea centralis. And this area contains the highest amount of cones, which helps for visual acuity. And this is usually where your eye will automatically kind of turn or angle itself to see an image so that it is reflected back on the fovea, because that will give it the sharpest vision. Okay, so I kind of went through function as I was telling you the structures with that. So the fibrous layer consists of the sclera and cornea. I'll let you guys kind of review these structures on your own as we go through there. The sclera just helps to give the shape to the eye. In the back, it's where your optic nerve exits. The cornea is the transparent part of the eye. It forms a clear window that light enters first, and it will bend the light as it enters into the eye. Numerous pain receptors contribute to blinking and tearing reflexes in the cornea. Then the vascular layer consists of the choroid, the ciliary body, and the iris. The choroid region is the posterior portion of the uvea, which is just this middle layer. It supplies blood to all layers of the eye, and it has a brown pigment, which absorbs light to prevent scattering of light. You don't need to know that. The ciliary body itself, anteriorly, the choroid becomes the ciliary body, which is a thickened ring of tissues that surrounds the lens. And your ciliary body consists of smooth muscle bundles. And that's important because it's smooth muscle, so it's involuntary. So this smooth muscle, ciliary muscles control the shape of the lens. And the shape of the lens, whether it's bulged out or flat, helps you read things close up or far away. So those are the ciliary muscles, which make up the ciliary body. And then we have suspensory ligaments, which extend from the ciliary processes to the lens to just hold the lens in position. The iris is the colored part of the eye. I have greenish, bluish eyes. Some people might have brown, blue, whatever, different colors of eyes. That's the iris. What the iris is, though, is the iris is also smooth muscle that helps to control the shape of the pupil. And the pupil is just the opening, the black part in the middle of the iris, but this opening regulates the amount of light going into the eye. In close vision and bright light, it causes the sphincter pupillae, the circular muscles, to contract, and the pupils will constrict. So in really bright light, the pupils constrict, and that's parasympathetic control. And then in distant vision and in dim light at night, your pupils will dilate, and the pupils will, the different muscles contract to cause the pupils to dilate or open up. And that's under sympathetic control. And if you remember, sympathetic nervous system gets your bodies ready for emergencies. So when you're in a fight or flight type situation, you want your pupils as wide, as open as possible to take in your surroundings and try to see everything that's going on around you. In parasympathetic, when you're resting, digesting after a meal, your pupils can constrict because they can kind of relax and they don't have to be taking in a bunch of light. This is also the reason if you've been drinking and driving and the cop pulls you over, he's going to check to see if your pupils will constrict when he shines a light in it, because when he shines the flashlight in your eyes, the pupils should constrict, because usually you drink and drive at night. Well, usually you should not drink and drive ever, but at night the eyes are dilated anyway, so causing the flashlight in the eye would cause the pupils to constrict. If the constriction with the flashlight doesn't happen, the cop knows something's up with the brain. So that's an easy way to tell of inebriation. Changes in emotional state can also occur. So pupils will dilate when the subject matter is appealing or requires problem-solving skills. So again, internal structures of the eye, you'll see a picture really similar to this, maybe one or two on your next lecture exam. So make sure you just are able to label these structures. This shows you which muscles contract for pupil constriction, that's the circular muscle, and which muscles of the iris contract for dilation. So again, the muscles that are contracting, whether the pupil's constricting or dilating, are in the iris. So your iris has two muscles that will contract for pupil constriction or pupil dilation. More structures of the eye, the inner layer is the retina. That originates as an outpocketing of the brain, because this is where we'll have all of our photoreceptor cells that transduce light energy. We'll have neurons, we'll have glial cells. We have an outer pigmented layer and an inner neural layer of the retina. And you can kind of see this here. So the pathway of light will travel into the eye through the cornea, past the lens, and then it hits the retina. The retina itself, you see the pigmented layer and the neural layer. We'll go through this in a later slide, but what light actually does is it goes and hits the back of the eye first, and then the nervous stimulation or the depolarization of the photoreceptors, the neurons, kind of make its way back and then out of the eye as the optic nerve. So that's kind of the way that light gets transduced into energy. It hits the back of the eye first, that pigmented layer, and then works its way through different photoreceptor cells, ganglionic cells, until the impulse gets transmitted through axons up to the brain via that optic nerve. So the pigmented layer helps to absorb light, prevents its scattering, it stores vitamin A. The neural layer is then the layer that runs just anterior to the margin of the ciliary body. It has a serrated edge called the ora serrata. You don't necessarily need to know that. And the different neurons that are in the neural layer are photoreceptors, bipolar cells, and ganglion cells. And the signals always spread from the photoreceptors, which are furthest in the back, and then they start to move forward to bipolar ganglion cells, and then the ganglion cell axons will exit the eye as the optic nerve. The optic disc, you should know that this is the blind spot of the eye because it does not have photoreceptor cells. This is where the optic nerve leaves the eye. And the retina itself has about 250 million photoreceptors that are one of two types. They can be rods or cones. And as we look here, this is another kind of picture of the retina. Rods themselves work in dim light. They're kind of your peripheral vision receptors. You have a lot more rods in your retina than you do cones. They're more sensitive to light, but they don't help with color vision or sharp images. And the numbers are greatest at the periphery of your eye to help you see in your peripheral vision, and again, in dim light. The cones then, these are the photoreceptors that help for bright light as well as high resolution color vision. So these help with your visual acuity, your color, help you see in bright light. The macula lutea is that area at the posterior pole, just lateral to the blind spot, which is where the optic disc is. And it contains mostly cones because the fovea centralis is a tiny pit in the center of that macula lutea that contains all cones. So it's the region with the best visual acuity, eye movement actually allows us to focus an object so that the fovea, again, can always pick it up. So even right now, as you're looking at my screen, or maybe you're watching TV while you're listening to me, or looking at your phone, your eye is constantly adjusting the image that's coming in so that it is centered onto that fovea centralis. So we're not going to go through specifics of how we see, but you should know that the pathway of light, again, goes all the way to the back layer of the eye first, the pigmented layer. So light kind of comes right in, goes all the way to the back, the pigmented layer, and then the signal of output travels back forward. And that signal itself will go through the rods and cones. And you can see here how rods, the kind of head of the rod looks like a rod, and the head of the cone looks more like a cone. And you have many more rods than cones in your retina. So the signal travels via action potential then through photoreceptors, rods and cones, through bipolar cells, which have just two processes on them. Remember, we talked about multipolar neurons, bipolar neurons. Well, here's an example of where we have these bipolar neurons. There's just two processes to them. And then ganglion cells, which will then have axons that go to the optic nerve. So through different we won't get into all the details. I kind of skip over that section here of how actually the signal passes through these different types of cells. But I want you to know the general idea that light hits the pigmented layer, and then the signal of that light gets translated or transduced into electrical energy as it travels through photoreceptors, bipolar cells, ganglion cells, and then out the optic nerve. This is a photomicrograph of the retina. Again, it's always fun to see a real life picture of these things. All right, the inner layer of the retina has two sources of blood supply, the choroid vessels and the central artery and vein that enter and exit in the center of the optic nerve. That these vessels are seen in a living person, and you might see this taken with an ophthalmoscope in the eye doctor's office. What they're looking for is they're looking at the retina to see if it's attached, to see if it's healthy. Here's the optic disc where the optic nerve attaches, and you can see the blood vessels entering the retina via the optic disc. So again, here's your blind spot because there's no space for photoreceptors there. And then just lateral to the optic disc, you have the macula lutea, that's the area. And the fovea centralis, which is not labeled, would be just a tiny pinprick in the center of that macula lutea. All right, a couple homeostatic imbalances. We have retinal detachment. This is a condition where the pigmented and neural layers can separate of the retina, and it allows that jelly-like vitreous humor to seep between them. This can lead to permanent blindness, and it usually happens when the retina is torn during a traumatic blow to the head or a sudden stopping of the head during movement, such as bungee jumping. I don't know if any of you have ever done that, but after this class, maybe you won't want to anymore. So that's retinal detachment, some sort of blow to the head. The victim of this retinal detachment describes it as kind of a curtain being drawn across the eye or spotlight, soot-like spots or flashes. Usually you can reattach the retina with laser surgery, but again, I don't know, just don't go bungee jumping or get into some sort of traumatic situation. Okay, the internal structure of the eye, a little more, we're almost done here. Again, we have the lens and the ciliary zonules, which are those suspensory ligaments that hold onto the lens, separate the eye into a posterior and an anterior segment. You should know that the posterior segment contains that vitreous humor, helps to transmit light. It supports the posterior surface of the lens. It contributes to intraocular pressure because it's more of like a jelly-like substance. And the vitreous humor forms in the embryo and lasts your whole lifetime. So the vitreous humor that you have has been there since you were in the womb. Anterior segment, the entire segment contains aqueous humor, which is much more kind of watery-like. It supplies nutrients and oxygens mainly to the lens, but also to the retina, and it helps to remove wastes. In the anterior segment, there's an anterior and a posterior chamber. The anterior chamber is between the cornea and the iris, and the posterior chamber is between the iris and the lens, but both are in front of the lens. So here are the different, this is the anterior chamber and showing just the circulation of aqueous humor, how it forms, and then it gets circulated up and into the anterior chamber. And you guys don't really need to know that, but it is interesting to see how the circulation goes into the kind of both anterior chambers of the anterior segment. And then you can see here the posterior segment behind the lens, which is the structure, contains that vitreous jelly-like humor. Glaucoma is a condition in which drainage of aqueous humor is blocked, and this can cause fluid to back up and increase pressure within the eye. Pressures could increase to dangerous levels to compress the retina and the optic nerve, and that could lead to blindness. There are a few early symptoms, but late signs could be halos around lights and blurred vision. You might get intraocular pressure tests where, I don't know if you've been at the eye doctor, when you get that puff of air at the cornea, which just freaks you out. That helps to measure any cornea deformation. And the treatment you can give some eye drops that increase the rate of aqueous humor drainage to decrease its production, or laser therapy or surgery. So the lens itself is biconvex, it's transparent, it's flexible, it's avascular. The lens itself becomes more dense and convex and less elastic with age. So that kind of leads to the idea that the older, don't want to say older, because some people in their 40s or 50s need reading glasses, and that's because their lens becomes less elastic. So it's not as able to focus on things close up. So that's why many of us will need reading glasses. Here's focusing light on the retina. So a little bit about the pathway of light entering the eye, which you should know. Let's notice my stars. Light enters through the cornea first, through the aqueous humor, the lens, the vitreous humor, the entire neural layer of the retina, and finally the photoreceptors. And light is refracted, and refracted means it's bent or focused, entering the cornea, entering the lens, and then leaving the lens. So light is actually bent or refracted 3 times. Most of the refractory power is in the cornea. However, it is constant and cannot change the focus. And what changes your focus is the shape of the lens, and that allows you to see things close up or far away when the shape of the lens changes. For distant vision, your eyes are best adapted for distant vision in general. The far point of vision is the distance beyond which no change in the lens shape is needed for focusing. And we call that at 20 feet for a normal eye, where the cornea and lens focus light precisely on the retina at this distance. The ciliary muscles are relaxed, which causes a pull on the suspensory ligaments, and the lens will be completely stretched and flat. So this just looks at the lens, and in distant vision, it becomes flat. And that helps you see distant vision, how the lens flattens for seeing things far away. You don't need to know, I don't think, what exactly happens to each of these during distant vision. For close vision, light from close objects, so less than 6 meters away, diverges as it approaches the eye, and this requires your eye to make adjustments using 3 simultaneous processes. Accommodation of the lens is the ability of the lens to change shape to increase refraction or bending. The near point of vision is the closest point on which the eye can focus. And presbyopia is the loss of accommodation over the age of 50. So the idea that you'll need reading glasses. I don't know why I said 40. I guess I've seen people 40 in the reading glasses. If you do have LASIK eye surgery, which is awesome. My husband got it when he was 18, when his parents had some extra money in their HSA one year. So he got LASIK. But if you do have LASIK, you most likely will need reading glasses, because if it corrects your far vision, eventually your accommodation will decrease. In order to focus on close vision, the constriction of your pupils, so accommodation pupillary reflex involves the constrictions of the pupils to prevent most of any divergent light rays from entering the eye. This is mediated by your parasympathetic nervous system. Convergence of the eyeballs, your eyeballs rotate toward the object being viewed. So this shows for close vision how the lens bulges or becomes fatter. And that's due when you contract the ciliary muscle, the ciliary zonules or ligaments become loose, and that allows the lens to bulge, and that helps you to see close. And again, in the elderly over 50, for accommodation, their ciliary muscles don't contract as well. So with that, the lens isn't able to bulge and see things close up. And then I think this is our last slide for this part 1 of chapter 13. I include this slide because these are problems associated with refraction or bending related to the eyeball shape. And we often get these names mixed up. So most of us who wear glasses are nearsighted. The myopia, when you were in 5th grade, like me, and you couldn't see the board, so you got glasses. That's the eyeball is too long, so the focal point is in front of the retina, and it's corrected with a concave lens. So that's nearsighted, is unable to see things far away, and hyperopia is farsightedness. It's unable to see things close up. And then astigmatism is unequal curvatures in different parts of the cornea or lens, and it's usually corrected with a cylindrically ground lens or a laser procedure. And if you have slight astigmatism, I do, you can kind of put kind of a corrective feature in your contact lens to help with that a little bit. But astigmatism, if you're looking at kind of a clock with the numbers and the clock hands going out, the numbers won't all seem the correct boldness. Some might seem a little blurry. So I'll stop the PowerPoint there, or the recording.